Today we are going to study about the analysis of aloe by chemical test. So the aim for the practical is to perform analysis of aloe by chemical test, that is aloe powder, and the requirements for the practical are test tube stand and the holder, beaker, funnel, pipette. So what
Now we will see the requirement for the practical, that is analysis of aloe powder by chemical test. Okay. So firstly we will require aloe powder, and these are the chemicals: bromine water, borax, ferric chloride, ammonia solution, petroleum ether, and the apparatus required are test tube with test tube stand and test tube holder, separating funnel, beaker which is filled with distilled water. Okay, we will require distilled water also. Pipettes are there, and spatula, and this is a test solution which I have already prepared. Okay, aloe powder was added to the water, it was boiled, then keselgar was added and it was filtered, and the test solution is prepared, and this solution we will use further for analysis. Now we will perform the first test, that is bromine test. Okay, we will take some test solution in test tube and we will add bromine water, and care must be taken while adding solutions to the test tube. Test tube should be filled, and we will mix it properly. Now you can see the pale yellow precipitate, okay, and this indicates the presence of aloe. Now we perform the second test. Okay, so for that we will take test solution, okay, which we have prepared already, and we will add borax to it. And we'll dissolve it properly. So we will mix it properly and allow it to dissolve. Okay, borax will get dissolved in the test solution. Till that time, we will take test tube, clean and dry, and we'll add some water to it. And now the previous test, borax with filtrate, and dissolve it properly. We will add this to the water containing test tube, and only few drops we have to add, and a green fluorescence will be obtained. Okay, this indicates the presence of aloe. Now we will perform the modified bond pair test. Okay, for that a test solution is taken. Okay, by adding aloe powder to the water. Okay, and now we will add ferric chloride solution to it. And dilute HCl. Okay, now dilute HCl is added. Okay, we will transfer this to the beaker and we'll add petroleum ether, or carbon tetrachloride is also allowed. As we have ether, so we will add ether to this. And we will transfer this solution to the separating funnel. Ether is added. We'll mix properly and we will add this solution to the separating funnel. Now we will shake it properly, okay, and while shaking we have to remove the stopper, otherwise it will bump out, and we will allow it to set. As the two layers will separate, we have to collect the organic layer, that is ethereal layer. Okay, so we will wait for some time and the two layers will get separated. Now you can see the two separate layers. Okay, upper one is the organic layer and this is the aqueous. Okay, and we have to collect this organic. Okay, now we will collect the layer. And while collecting, you have to keep the separating funnel open. Okay, no stopper should be there. This is the aqueous layer which is of no use, and now we will collect the organic layer. And to this organic layer, okay, this is the organic layer, we will add ammonia solution, okay. See, ammonia is added, okay, and now we will add this solution to the separating funnel again, okay. You have to check that the knob of the separating funnel is closed, and we will add. And from this, the ammonical layer has to be collected. Okay, now we will allow it to settle for some time, and the organic layer will be upwards and the ammonical layer will be downwards, and to which pink color or rosy red or rosy pink color will be obtained, okay. Okay, the layers are separated, and now we will collect the ammonical layer, and you can see red pink color, okay. For clear visualization, I will transfer this to the test tube, and here you can see, and this indicates the presence of aloe, and this is the taste modified bond agar test which is performed for aloe
and bonds.